Breaking the Wall of Molecular Imaging in MRI. Ursula Pfeiffer, Leibniz Institute for Molecular Pharmacology, Germany.
Welcome to Berlin. For a start, I'd like you to say, on the count of 3, here I am. 1, 2, 3. Here I am. Thank you. This is how an MRI, a detailed overview in any point of your body is generated by looking at the vast population of hydrogen nuclei. Now, to tell healthy and diseased tissue apart, it is very hard to do with just looking at the hydrogen nuclei, which is why we use contrast agents. But even with modern contrast agents, tumors are only detected when they're already a few centimeters thick. And this is the wall I want to break today, between a few centimeters and single cells. Many people like I are looking for molecular markers for this technique, for rare marker molecules. But whatever signal we will get from those will be hard to find. For instance, calcium. Calcium is lowly concentrated in living tissue, but the calcium level will tell us a lot about the state the cells are in, and is an indicator of several diseases, amongst those cancer, for instance. The cell has its inbuilt calcium sensor, which I am using. This protein, called calmodulin, wraps tightly around its binding partner, thereby changing its state, its structural conformation, as it senses calcium. And by changing the state of this binding partner, it will also change the state of a contrast agent attached to it. And this is what I'm using here. To generate contrast, I'm using a technique called xenon hypersest. I'm doing a xenon MRI instead of a hydrogen MRI. And with this particular construct of my binding partner of calmodulin and the xenon sensing agent, I'm onto something really good, because the negative contrast is not only very strong, it is also switchable. Meaning, this molecular hug that I described earlier is a switch-on effect for the contrast. So when there's no calcium, my response is silenced. Only when enough calcium is present beyond a certain threshold, calmodulin will sense it, perform the molecular hug, switch my contrast agent on, and I can generate negative contrast, which is highly robust. And just to give you an idea of how this would look in an image, because you see spectra here, so you have a two-compartment glass vial here with water on the inside and a xenon exchange agent on the outside, which lights up as a result of this negative contrast. And if you do not know what to ask me, please ask me what xenon hypersest is. So thank you very much.